Okay. All right, AP class, it's Tuesday. This is the day at this point, will probably be at the zoo surrounded by a bunch of 6-year-olds. So, sorry I missed you, but I think it's a good opportunity for me to try to utilize technology to keep moving forward. So, the one thing I want to do here, first of all, today is just review what hopefully we covered yesterday in class on Monday. We finished up talking about the details of meiosis, okay? But I think it's important. I love this slide because it shows you major differences. It's a comparison of mitosis, which is on the left-hand side, and meiosis, which we just recently covered. So I want to kind of go through this step by step. I'm going to start with mitosis, and so you can kind of use this as a review of mitosis, and we'll talk about at the end how they're similar and how they're different. Okay, mitosis. The parent cell for this example is a parent cell that has 6 chromosomes. So 2n equals 6. That's its diploid number. So the parent cell has 6. Now remember, humans have 46. So obviously, this parent cell, or this organism, is not human. That cell is going to undergo chromosome duplication even before it enters mitosis. It's going to do that in the S phase of interphase. So they basically, that's why they have the chromosomes looking different there than here. And by the time they get here, those chromosomes have duplicated, DNA replication. They're still considered to be 6 chromosomes, but now they're in this sister chromatid form. And in mitosis, remember, it's basically, it starts with prophase. In the prophase process, that's basically when you're going to get ready for these chromosomes to divide. The nuclear envelope's going to disappear. The mitotic spindle's going to start forming. During metaphase, all of those chromosomes, in this case there's 6, are going to line up in the middle during metaphase. But they're going to line up single file. That's very important. That's a huge difference from here to what we'll see next. The chromosomes line up in their sister chromatid form. They all line up in the middle, single file. 1, 2, 3, 4, 5, 6 chromosomes. Then during anaphase and telophase, and they kind of go through the process real quick, those sister chromatids are going to separate from each other. The cell will eventually become daughter cells, and what's important here, they didn't write the word cytokinesis, but I wish they would have, but what's really important here is that you are creating cells that are diploid from this process. In other words, you start with a diploid cell, you end with 2 diploid cells. Same number of chromosomes there as you see here. Genetically identical chromosome content, genetic material there, genetically the same here. So mitosis makes genetically identical cells, same number of chromosomes. Meiosis, okay, there's a lot more terms associated with this process. You're still starting out in this case with a parent cell with 6 chromosomes. That cell will still undergo, those chromosomes will still undergo duplication in interphase, of the S phase during interphase. However, a couple of things that are different here. Meiosis is a 2-part process. You have a meiosis 1 and a meiosis 2. So it's 2 phases as opposed to just 1 over there. Each of those phases is going to have its own prophase, metaphase, anaphase, and telophase. So you're going to go through the 4 of those phases twice. There are some other differences. When it comes to meiosis, the first part, prophase 1, you have something really important going on there called crossing over. You also have a situation where, and then there's some other terms associated with that. You have this chiasma, which is basically referring to the place where those, chromosomes are going to hook up. You also see that they're paired up here. You didn't see them pair up over there. They are paired up over here. That's what this term homologous chromosome means. A homologous chromosome is a paired up chromosome One chromosome came from mom, one chromosome came from dad. Okay, so there are chromosomes that are coding for the same genes. They could have multiple forms of that gene depending on if moms and dads were different, but they're coding for the same gene. Homologous chromosomes, a pair of chromosomes originating from mom, one of them, and dad, the other one. So that what is really important in meiosis 1 is that they're going to pair up together. Okay. Metaphase 1, they're pairing up together. They've already done this thing called crossing over in prophase 1, where a little bit of the genetic material was flip-flopped between them. Now they pair up, or now they line up. They're not single file here. They are not single file like we saw over there. They're paired up. Kind of fast forwarding here, by the end of meiosis 1, you've gone from a diploid cell with having 6 chromosomes up there to having a haploid cell with half the number. In other words, during the first phase, meiosis 1, we've reduced the number of chromosomes in half. Now, but at this point, we've only made 2 new cells. We're not done yet, though. But we've already reduced the numbers in half. Okay, 6 to 3. This meiosis 2, the second part, is very, very, very similar to what we saw over there. So these 3 chromosomes, for example, are going to line up in the middle, single file, undergo that, that in anaphase, the sister chromatids will separate, and bam, you'll have 2 new cells from this cell, 2 new cells from this cell. So, at the end of meiosis 2, you have 4 haploid cells. Crossing over has resulted in those chromosomes having exchanges of genetic material, which is why you see red and blue all over the place in different places. But you start off with 1 diploid cell, you end up with 4 haploid cells. Okay, so to kind of summarize, the big differences. In both mitosis and meiosis, you have DNA replication occurring in the S phase, so even prior to cell division. That happened both. So chromosome duplication, S phase. Okay, that's the same. Mitosis has just 1 set of divisions. Meiosis has 2 sets of divisions called meiosis 1 and meiosis 2. In mitosis, you don't have anything like crossing over. You do not have homologous chromosomes pairing up. Over here you do. You have that process of crossing over. You have homologous chromosomes pairing up. The overall result of both of these is different. In mitosis, you end up with 2 diploid cells. In meiosis, you end up with 4 haploid cells. The whole point of a cell undergoing mitosis is for growth and repair. And sometimes asexual organisms will reproduce that way. The whole point of meiosis is far different. It's just to make gametes, so it's reserved just for those cells that undergo sexual reproduction. This is a major part of that player. So that's kind of a breakdown of what we talked about yesterday, a little bit of a review. Make sure you understand those processes really well. Right now, I actually kind of want to shift gears and talk about the last thing that's talked about in this chapter. So kind of shifting gears here. The last part of your chapter kind of sums things up. They sum things up. They kind of talk about, they kind of review a little bit, but they kind of make it into a big picture. How genetic variation is achieved. Okay, we mentioned earlier in this chapter that it's really a good thing to have genetic variation. You want cells to undergo genetic variation because it's much better for them in the long run when it comes to changing environment. Okay, so the things I'm going to be talking about specifically here are basically, initially at least, are specific for sexually reproducing organisms. In other words, this, we're going to be talking about ways that you get genetically different cells, but this is only going to refer to those organisms that undergo meiosis as part of that process, in other words. Okay. Something called independent assortment helps attain genetic variation. Something called crossing over, which actually we've already talked about, but it's good to hear it again, is very helpful to result in genetic variation. And then something called random fertilization, which is actually an extremely easy concept to understand, is definitely another reason we have genetic variation. In other words, you can kind of think of it this way. Together, these things result in you not looking like your parents. Okay. So think of it that way. Let's start with independent assortment. You have to remember, to understand independent assortment, you have to remember that homologous chromosomes are going to pair up during meiosis 1. During prophase 1 of meiosis 1, homologous chromosomes are going to pair up. However, independent assortment takes this one step further and says, yes, those homologous chromosomes are going to line up, but they will also line up independent of each other. Okay, they're going to line up independent of each other. So what does that mean? That means, and I'm going to simplify this, so I'm not going to draw out, I'm not going to refer to a human here, but let me get another colored marker. Let's say I have a cell that only has 4 chromosomes. Okay, I have a cell. I'm going to simplify it so it may not be perfect, but I have a cell that has 4 chromosomes. I'm going to make the homologous chromosomes different colors. So this cell has 4 chromosomes. Obviously, this cell's about ready to undergo, it's in the prophase 1 phase of meiosis, so they're paired up. They're getting ready to the homologous chromosomes are getting ready to separate. But what this is saying is, you know what, independent assortment, yeah, there is a chance that the homologous chromosomes will line up this way, but there is also a chance that the homologous chromosomes will line up this way. Oops, let me make that black. Oh, I'm going to number these. I'm going to number these. This is going to make it easier. In other words, there's a chance that they'll line up like that, but there is also a chance that instead of chromosome number 3 here being on the left-hand side, chromosome 3 might actually flip itself around this way. Not sure I color-coded these right. Okay, let's see if we can see this. All right, here's what this, oh, this is a terrible picture. Here's what this is saying. The chromosomes might line themselves up. This is chromosome number 1 right here. This is chromosome number 2 right here. This is chromosome number 3 right here. This is chromosome number 4 here. However, independent assortment is saying, you know what? 3 and 4, they might have actually lined up, flip-flopped of each other. So instead of having the 3rd chromosome here, that chromosome 4 might have been here, that chromosome 3 might have been over here. So what does that tell me? That tells me that when these homologous chromosomes are going to separate, and they will eventually, chromosome 1 and 4 are going to end up with each other here. Chromosome 2 and 3 are going to end up with each other here. That's different from up above. Up above, chromosome 4 and 2 connected with each other. Chromosome 1 and 3 will end up with each other. So independent assortment is saying, you know what? Homologous chromosomes are going to line up independent of each other. It doesn't matter. So they could get some of mom's original chromosome and some of dad's original chromosome. They just can line up independent. Now, obviously, this is just an example using 4 chromosomes, but think about a human cell that has 46 chromosomes. That's a lot of different combinations just by lining up these homologous chromosomes and flipping them around. They're lined up independently. Okay. By the way, there's a little math involved here. There is actually a way to determine the possible number of combinations that can arise based on how many initial chromosomes that organism has. Now, for an organism that only has 4 chromosomes, there's not going to be nearly as many combinations that can arise as there will be for a human, for example. Here's how you figure that out. This is the magic formula you kind of have to remember, 2 to the n. In other words, if you want to determine how many different ways chromosomes can line up, you use this formula. Okay. In this particular formula, you started off with a cell that had, oh, by the way, the N, before I go into the detail, that N, which you should already kind of see, or already kind of remember, is a haploid number. Okay. So in order to do this math, you have to figure out what the haploid number is for that. So you got to kind of be careful in that sense. Okay. Using the example above, simple example. This chromosome, it started off with a 4 chromosome organism. That's the diploid number. In order to do this problem, I need the haploid number. The haploid number would be 2. So 2 to the 2nd power is going to be what? 4. In other words, there are 4 different combinations that will result in different cells just from this process alone. Now let's talk human. Humans, our haploid number is 23. If I did the math on this, and I wanted to figure out how many combinations that can result from the homologous chromosomes pairing up independent of each other, you can get a calculator and check this math, but 2 to the 23rd power is 8.4 million. In other words, there are 8.4 million different ways those homologous chromosomes can line up with each other. And that's simply because we have a lot more chromosomes than the picture here. Okay. All right. That is independent assortment. And that's one reason we don't look like our parents. Okay. Crossing over. We actually already talked about this, but I want to introduce a new word. Results in recombinant chromosomes. And that's just a fancy word for, as a result of crossing over, you're kind of making new chromosomes. You are making new versions of chromosomes with genes from 2 parents or from both parents. In other words, I won't go into detail because we already talked about this a lot. As a result of crossing over, you're exchanging genetic material between chromosomes, almost on a gene by gene basis. Just kind of for the record, if you took a different chromosome, or if you took each of your chromosomes, typically in a human, when your cells are undergoing meiosis and crossing over is happening between two homologous chromosomes, there is about 3 crossover events per chromosome pair. In other words, there's going to be 3 separate points that genetic material is going to be exchanged along those homologous pairs of chromosomes. That's just an average number. It's just kind of interesting. In other words, you're going to be resulting in brand new chromosomes, far different from mom, far different from dad as a result of crossing over. So that in combination with independent assortment, the number of different gametes that you can even make is phenomenal, is some outrageous number. Because not only can those homologous chromosomes line up differently, they're not even going to be the same to begin with because of crossing over. They're not even going to be the same as they were from the parent to begin with because of crossing over. So again, these two things result in you looking far different from your parents. Okay, random fertilization. You know what this refers to? It's actually very easy. Look down here. When you make, this is what I'm going to do. This might be a little kind of difficult to understand. When a female cell produces eggs, as we already did the math, it can produce eggs that have basically 8.4 million different combinations of chromosomes just from independent assortment alone. In other words, when females produce eggs, those eggs there's 8.4 million different combinations that can result from that process. And that's just independent assortment. That's not even taking into consideration crossing over. I won't even go there with the math. That's too complicated. In order to make a sperm cell, or sperms, there are 8.4 million different combinations that can result per individual for making a sperm. In other words, your egg and your sperm are obviously, there's a lot of different combinations of those that can form. Now, what random fertilization is saying, if that individual is capable of making 8.4 different combinations, and this male is capable of performing 8.4 million combinations, the chances of one sperm and one egg it doesn't matter how many times that individual makes sperms or eggs, they're basically going to be a much more, in fact, if you did the math, what you would actually end up doing is taking 8.4 million times 8.4 million and coming up with this number 70 trillion. What I'm coming with, this number that I just have, is kind of hard to explain, but basically I've just given you this really super large number by multiplying those other two numbers. It's like a probability. It's basically a number of combinations. A random egg and a random sperm are going to hook up, but it's never going to be the same egg and sperm that are going to hook up. So if this sperm up here is just one of 8.4 million that were made by that male. This egg right here is one of 8.4 million that were made by that female. The chances of that same genetic combination sperm that was made to begin with hooking up with an egg that is also the same genetic information that was to begin with is never going to happen. random fertilization. In other words, that sperm it'd be very rare for two sperm to actually have the same genetic combination to hook up with another egg with the same genetic combination. I know I'm not making any sense. I will clarify tomorrow when I see you in person. Okay. That's what I wanted to talk about there. In other words, these three things are huge when it comes to producing genetic variation in an organism. And again, remember, these things are all very specific to organisms that reproduce sexually, okay? However, then the very last thing your book talks about is, hey, don't forget those asexual organisms. And you're like, well, wait a minute. I'm saying, okay, so don't forget the asexual organisms, but wait a minute, Mrs. Hatcher, I remember, you're producing, when asexual organisms reproduce, you're making a genetically identical cell. Well, yeah, the actual process, like binary fission, produces a genetically identical cell. Or if an organism undergoes mitosis to reproduce, yes, you're producing a genetically identical cell. But we've said that having a genetic diversity has an evolutionary advantage. In other words, organisms actually are going to adapt better to a changing world if they are genetically diverse. So there's this rotifer. It's a protist from the kingdom protist. This thing has been in existence for 40 million years. They can see that in the fossil record. Okay. They know that this rotifer reproduces asexually. Probably very similar to a mitosis kind of process since it's a protist. Okay. They're like, well, wow, how has that thing managed to live for 40 million years but reproduce asexually? And what they've seen is, you know what? Even though this protist reproduces asexually, there's actually exchanges of genetic information prior to the reproduction process. There's a technique called horizontal gene transfer. By which DNA is going to be transferred from one rotifer to another by junctions. In other words, when it comes to actually reproducing another rotifer, these rotifers are actually going to reproduce by asexually, but prior to reproduction, they're actually exchanging genetic material a different way. By basically horizontal gene transfer, by hooking up with another cell and exchanging genetic material between the two of them, resulting in combinations. This is why they think that rotifers have like the best of both worlds, these rotifers, this specific breed. They not only reproduce asexually, which is quick, which is energy efficient, but they're also able to get a little bit of genetic variation, enough to keep them adaptable in a changing environment. It's like the best of both worlds. Okay? Again, we did briefly talk about bacteria. Remember, bacteria also produce asexually. They do the same thing. They're able to reproduce asexually, so it's quick, it's energy efficient, all that. However, bacteria also undergo a process very similar to horizontal gene transfer, but it's called something different. It's called conjugation, but it's basically very similar to horizontal gene transfer, where these bacteria are going to get together and transfer genetic information between each other. So really, they're going to be genetically diverse. And then those cells will go on to reproduce asexually by binary fission and produce new bacteria. So again, it's like the best of both worlds. They're reproducing asexually, but they're still changing genetic material. So, and I think that covers the end of the chapter. I'm going to kind of keep it brief for today. Oh, you know what I might do? Let me show you some pictures from your book that go along with what we just talked about. And it might make a little more sense out of that random fertilization too, once you see it in a picture. I don't think I explained it very clearly at all. Not a problem. Okay. Again, the big things, oops, let's wait here. The big things we talked about today were these 3 ways that genetic variation is achieved in organisms that reproduce sexually. And the first thing I talked about, and what this slide is going to show you, is that independent assortment idea. Okay. You know what? Their colors are probably better than the ones I used. I numbered my chromosomes. They actually color-coded them. They did a better job of it. So this is another way of viewing it. 2 possibilities in this case of ways that those homologous chromosomes can line up with each other. I like the way they did it better. In other words, you have, this is a 4-chromosome cell. Those homologous chromosomes are pairing up, and they can pair up in that way, where you have a blue chromosome on both sides, they color-coded them. Or the homologous chromosome, the chromosomes on the bottom, you kind of have to take this pair right here and just do a flip-flop of them. In other words, they can pair up that way as well. But whatever the case, they're going to result in different gametes being produced. And the possibility number 1, the 2 chromosomes that are blue found their way into a cell. The 2 chromosomes that are red found their way into a cell. But because they lined up different over here, a blue and a red ended up together. Just by chance. Then they just took it through the end of meiosis and said, as a result of just the homologous chromosomes lining up different, you can produce these 4 haploid cells, or you can produce these 4 haploid cells. Very important picture to kind of look and understand. This is independent assortment. Has to do with how the homologous chromosomes paired themselves up, or how they lined up, independent of each other. Okay, this picture is showing you the details of crossing over. Okay, so you have a blue and a red at the very top. You have these homologous chromosomes, 1 blues, 1 red. They are going to attach at certain areas along the chromosome called the chiasma. That's going to result in exchange of genetic material. So a little bit of this chromosome ended up over here and vice versa. You have exchange of genetic material between homologous chromosomes, which is going to create the possibility of gametes to be even greater. You're flip-flopping individual genetic information on a chromosome between them. Again, another reason we don't look like our parents is because of this crossing over process. That they just show you a detailed picture of a real cell undergoing crossing over. That point where they attach is called the chiasma. I don't know why they labeled the centromere. There's just to show you that's the center of the chromosome. I guess to show you that crossing over doesn't have to happen at a centromere. Oh, this is a picture of that rotifer that reproduces asexually, but is still able to oops. Oh, these are just other pictures. Okay, this is something we hopefully probably covered yesterday in class. Okay, that's all I have for today. I'm going to end it there. Hey, remember tomorrow we are going to do a lab. So if you get a chance, please read the photosynthesis lab in your AP lab manuals. That'll make the lab tomorrow go much more smoothly. And again, remember Thursday test. At this point, we've covered all the information from that will be covered on the test. So if you have any questions about anything, we can answer them during lab on Wednesday, definitely, or email me, and we'll figure them out. So, alrighty, guys, I think that's all for today. Enjoy.